Welcome back. We're going to finish talking about the somatic nervous system when we talk about the control of movement. And I think one aspect that's interesting about this is how much of it is really focused in the spinal cord versus in the brain, and that will become evident in a few minutes. First, we're going to talk about the fact that there are actually two types of lower motor neurons, meaning the motor neurons that are innervating the skeletal muscle. There are alpha motor neurons. These are the ones that we're used to discussing. These are the ones that are innervating what we call the extrafusal muscle fibers or cells. These are the fibers of the main part of the muscle, and these are the ones that are doing the contracting to cause the muscle to produce tension. So these are the main muscle cells, and the alpha motor neurons are the ones controlling those muscle cells. So this is what's doing the work of a muscle, are the alpha motor neurons activating the extrafusal muscle fibers. In muscles, we also have another structure called the muscle spindle, which is consists of what are called intrafusal fibers. That's what are shown here, which are special muscle cells that shorten, but they don't really generate force of the muscle. Another component of this muscle spindle, which is contained in this capsule right here, is our afferent neurons that are going to sense the stretch of these intrafusal fibers. And so it's going to be gamma motor neurons that are going to innervate these intrafusal fibers. So you might ask, okay, if these intrafusal fibers aren't contributing to the force producing capacity or ability of the muscle, why do we have them? And so the point of this muscle spindle is to have this parallel system of intrafusal fibers and this parallel system of the alpha and gamma fibers, so that whenever the alpha fibers or the alpha motor neurons are being stimulated to cause muscle to contract, the gamma motor neurons will also be stimulated, so that as a result, whenever the extrafusal fibers shorten, the intrafusal fibers should shorten in the same manner. And so in this way, the muscle spindle is like this parallel system that is special because of this afferent neuron sensing the stretch in the fiber. And so it's this parallel system that lets the body monitor the length of the muscle by monitoring the length of the intrafusal fibers, which are activated in the same manner and at the same time as the extrafusal fibers. And so this is going to be one of our aspects of muscle length that is going to be monitored for proprioception, is the amount of stretch. And so this is going to be critical, this muscle spindle is going to be critical for monitoring the length of the muscle as it shortens, so that the more it's stretched, the more firing there will be of this neuron. And then as we contract, the firing will reduce less and less, and that will tell the central nervous system that the muscle is getting shorter, and because there's less stretch, so there'll be fewer action potentials, and then that will tell the body about where, for instance, the position of the hand is based on how short the muscle is. So these alpha and gamma motor neurons are going to be efferent somatic motor neurons, and they're going to be co-activated so that this muscle spindle will reflect what's happening in the rest of the muscle. We're going to switch gears just a little bit to now talk about muscle sensory receptors, and you've probably guessed we've already talked about one of them, and that's going to be the muscle spindle. So it's going to be an important sensory receptor for muscle. Is going to be the muscle spindle. And so you can see how the afferent neuron that we're just talking about, it's coiled around the muscle, and it's going to sense stretch. You can remember it because Spindle starts with an S, and so does stretch. And that means that when the muscle is stretched, that's when the neuron is firing the most, and then as the muscle shortens, it's going to be firing less and less. When it's firing a lot, because there's a lot of stretch, it can cause a reflex that is going to cause contraction of the stretched muscle. So this is going to be a negative feedback loop, which means that if we're saying the problem is that this muscle is overstretched, then to correct that problem, it's going to want to cause contraction, for instance, let's say, of the bicep. Now, to do that, it's also going to need to worry about what's happening with the tricep, because if you want to shorten this muscle, the bicep, then you're also going to need to relax the tricep. And so the circuitry, and we'll see the specific circuitry in a minute, is going to cause what we call reciprocal innervation, which means we're going to cause contraction of the stretch muscle, and then we need to cause the opposite action, relaxation, in the antagonistic muscle. So that would be the bicep versus the triceps. So in this way, that gamma motor neuron that is going to activate the intrafusal fibers to cause them to shorten alongside the extrafusal fibers, and the muscle spindle, which is the sensory portion, are going to act in this negative feedback loop so that we can keep the muscle length at a desired value, and we'll see how that happens more in a minute. Our other muscle sensory receptor is going to be called the Golgi tendon organ, which is going to be located in the tendon that's attaching the muscle to the bone. And so this is going to respond to a different type of stimulus. We said the muscle spindle is going to be activated when you stretch the muscle. When you stretch a muscle, the Golgi tendon organ, or the tendon itself, is not going to be stretched that much. Because when you stretch a muscle, the muscle is going to give, so that there's not that much tension put on the tendon. So if the Golgi tendon organ fires very infrequently, just when the muscle's at rest, when it's stretched, when the muscle is maximally stretched, the Golgi tendon organ might fire a little bit more. But when it really fires, and remember, these are going to be mechanoreceptors in this afferent neuron that's going to respond to tension in the tendon. When that Golgi tendon organ is really going to fire is when you're maximally contracting your muscle, because that's when that tendon is pulling against the load, and it's kind of caught between the contracting muscle and the load. And that's when the Golgi tendon organ is going to just fire like action potentials like crazy. So that's why we say the Golgi tendon organ is responding to tension in the muscle, not stretch. And I always remember it because tendon of the Golgi tendon organ starts with a T, just like tension. So the Golgi tendon organ is sensing tension generated by the muscle, which is basically going to be when it's contracting. It's going to be an afferent nerve ending located at the junction of the muscle and the tendon, and then we're going to have an afferent axon. So it's going to be a mechanoreceptor stretch activated ion channels in this Golgi tendon organ. So that means that since Golgi tendon organ is activated by contraction, then they're going to act to negatively feed back to regulate tension, so that if the Golgi tendon organ is really firing because you're lifting a really heavy load, it's going to act to reduce contraction, to relax the contracting muscle. And then again, it's going to have to act with reciprocal innervation to then cause contraction of the antagonistic muscle. So it's going to act to fix the problem, to use negative feedback, but it's going to act to relax the muscle that's contracting. And we'll see a more concrete example of this in a moment. So let's talk about reflexes. where we're going to have some sort of sensory input. Then the central nervous system is going to decide what to do about that input. And then it's going to cause a response that's going to be controlled by an efferent pathway like the somatic nervous system. We're going to talk about first the muscle stretch reflex, which is basically an example of this would be the knee-jerk reflex, when the doctor might use a little hammer as you're dangling your leg over the examining table. And when he does that, he is striking the patellar tendon under your kneecap, which is then tugging on the kneecap and then on the quadricep muscle. And it's basically causing the quadricep muscle to stretch really quickly. And so that's going to activate the muscle spindle. And we know that when we activate the muscle spindle, that's going to cause contraction of the muscle and relaxation of the antagonistic muscle. And that's what we're showing here. So we've activated the spindle. That's going to cause contraction of the quadricep muscle and relaxation of the hamstring muscle. And we're going to do that through the action of an inhibitory interneuron that's sitting between the muscle spindle neuron, afferent neuron, and the neuron for the hamstring. So we have an inhibitory interneuron, and it's going to release a neurotransmitter such as GABA or glycine, very commonly, in a synapse with the somatic neuron for the hamstring. And those neurotransmitters often cause opening of chloride channels, which is going to cause chloride to rush in to the somatic motor neuron controlling the hamstring. And if chloride rushes in, negative charges rush in, and those channels stay open, it's going to be much harder to cause an action potential in that neuron. And if you have a reduction in action potential frequency, that's basically relaxation of a muscle. So again, our language here is action potentials. So when you want a muscle to contract more, you have a higher frequency of action potentials. If you want it to relax, then you greatly reduce the frequency of action potentials. And so when we're talking about antagonistic control or reciprocal innervation, we're causing one neuron to fire more rapidly to cause one muscle to contract, and then we're causing the other neuron, leading to the antagonistic muscle, to fire less frequently, which is going to cause relaxation. And so that's what's happening here when we have this reflex, where we've got activation of the muscle spindle, activation of the quadricep muscle, and then activation of the inhibitory interneuron, which prevents firing of the neuron that is leading to the hamstring. Now this, obviously, the vast majority of our movements are not completely reflex. One thing to note with this is that this reflex loop that we're talking about really involves only the spinal cord. These are the neurons, and this is only involving the spinal cord. However, we can use reflexes like this by influencing them from up above, from the brain, to do more everyday tasks. So an example of using this muscle stretch reflex would be if you're holding a glass for someone to pour a drink into. Your brain knows that if you're holding this empty glass and someone's about to pour a drink, you need to keep your arm level so that when they start to pour the drink, you don't just dump it because of the extra load from the drink. And so what the brain can do is say, okay, this muscle is right where it needs to be. I don't want it to get any longer. I don't want it to stretch anymore. And so it changes the sensitivity of the muscle spindle so that now it's not only going to act just if it's completely stretched in a dramatic way. it's going to act and be super sensitive so that it's going to cause a reflex if this muscle gets longer at all. And so what will happen is you've got this new amount of sensitivity set into the system from the brain affecting this same circuit we've just been talking about. And someone starts to pour a drink, the load gets greater because this is heavier, so your arm dips just a little bit. But that's when that muscle spindle says, no, I don't want to stretch. And so it causes the same reflex that we just saw with the knee jerk reflex, where it's going to cause contraction of the bicep and relaxation of the tricep to keep this drink level, keep your arm level so that you don't spill the drink. So in this way, the basic circuitry is the same as this completely reflexive action of the knee jerk reflex, but we are able to modify that system by the brain, through the brain, and use it for a more everyday purpose. So let's talk about the Golgi tendon reflex. That's also going to cause a reflex, where we said, remember, the Golgi tendon is going to be really activated when the muscle is really contracted and really pulling dramatically on that tendon. So that means we've got the signal coming from the Golgi tendon organ, coming into the spinal cord. Its cell body is going to be sitting here in the dorsal root ganglion of the spinal cord. And it's going to act on an excitatory interneuron in the tricep to then cause excitation of the somatic motor neuron leading to the tricep to cause contraction of the tricep. And then it's going to act to activate an inhibitory neuron, which will then inhibit the neuron leading to the bicep to cause it to relax. So it's, again, trying to fix the problem of too much tension in the muscle, and it's going to do that by relaxing this muscle and contracting the opposing muscle. So it's going to act with reciprocal innervation again, and this is going to act to limit the tension within the muscle so that we can protect the muscle and the muscle tendon junction so that they don't damage or just rip apart. We can also have influence of a stimulus that is not in the muscle itself or not in the tendon itself. It could be, for instance, a pain stimulus coming from the skin. We can also cause a reflexive action that involves the somatic motor system. And this is an example where it's called the withdrawal reflex, because in this case someone has just stepped on a jack, and as a result the reflex is going to be to withdraw the foot. So that means that the quadricep is going to be relaxed and the hamstring is going to be contracted to do that withdrawal. Now if you withdrew your foot and didn't make an adjustment in the opposing foot, you would just fall right over. And so this withdrawal reflex not only involves withdrawal of the limb, but it also involves the crossed extensor reflex, which causes the opposite action in the opposite leg, where instead of withdrawing the other leg, you extend it, which allows you to support all of your weight on the foot that's not experiencing the pain. So this gets even more complicated because not only do we have reciprocal innervation in each leg, but we also have opposite actions in each leg. And so that's what this animation is showing, where now we're activating to withdraw this leg and to extend this leg through, again, afferent pathway, this time from a pain receptor. And so we're getting reciprocal innervation of each leg and causing opposite effect in one leg versus the other, to withdraw one leg and extend the other leg. And again, this sort of reflex can be modulated by the brain. So if you pick up a hot plate, your reflex is going to be to withdraw your hands However, if that plate is really precious china, or it's got your favorite meal on it, you might be able to kind of tell yourself, okay, I know it's hot, but hold on. If it's too hot, you might not be able to do that. But if you do have some control in some instances over your reflexes, and again, it's just the brain influencing these very circuits that we're looking at. That's going to move us to talking a little bit about central pattern generators, which are going to be these spinal cord centered circuits that are going to be responsible for a lot of our repetitive movements that happen in everyday life. So these are going to be neural circuits that are going to coordinate complex patterns of movement, and that it's going to be independent of sensory input to some extent, but that it can be adjusted in response to sensory feedback. And I'll give you an example. So because these are repetitive movements, that means that these circuits that are generated are going to be oscillatory. And they are going to be flexible due to control by the brain. So let's talk about one of the easiest things to see as an example of this would be walking, where you've got two different actions that you're going to do, two main actions that you're going to do. One is that you're going to have the swing phase when you walk, which is going to be whenever your foot is off the ground. You're swinging it from being behind you to in front of you. So that's the swing phase. Then the stance phase is when you've just stepped on the foot, and then you're going to push to move your body back, to move your body forward. That's called the stance phase. And so each leg is going to alternate between being in the swing phase and the stance phase. And again, you're going to have reciprocal innervation of the different muscles to allow for basically withdrawal and extension. But then when your right leg is in the swing phase, your left leg will be in the stance phase. And so again, like with the withdrawal reflex, you're going to have reciprocal innervation of the limbs as well. So it's actually somewhat simple or very repetitive motion where you're just repeating the same sets of muscle contraction, but you need to make sure that the left leg is doing the opposite of the right leg at all times. And so this can be established and based in the spinal cord through these central pattern generators, where they just generate this pattern of neural activity that causes you to walk. This can be adjusted by the brain, and that's adjusted in response to sensory feedback and influenced by the brain. And example of that would be if you want to go from walking to running. It's the same general pattern. It's just you're going to reduce the stance phase, and that will allow you to run. So it's just a little modulation of the main central pattern generator that allows you to switch to running. And it's efficient because you get to use the same basic circuits and just modify them to do a different activity, such as running versus walking. This has also been pretty well studied in quadrupeds, so in animals that have four legs. When, for instance, a horse goes from a trot to a walk to a gallop, they still have a similar motion of basically a swing versus a stance phase with their leg, but the pattern of which leg is in which phase changes depending on how their gait. And so again, it's a modification of one of these central pattern generators that allows for that different pattern of movement in different types of gaits in a quadruped animal. So we've talked about our sensory receptors in muscle that are going to regulate muscle length and velocity of shortening and tension in the muscle. And remember how we can use these in terms of just being able to monitor what's going on with the muscle in terms of proprioception, but then also in using it as a reflex to protect the muscle or to do everyday activities as well. And then we do have some reflex actions that are involved with any sort of sensory information. We talked about sensory information coming from muscle, tendons, or skin and causing a movement in an appropriate muscle. And then we talked about central pattern generators and how they can be important in everyday repetitive movements such as walking, and how it's going to involve having antagonistic muscles being reciprocally innervated as well as reciprocal innervation of certain limbs as well. So central pattern generators will be involved in many everyday processes that are repetitive in nature.